Every time you consume a meal, your digestive system metabolizes carbohydrates into pure energy. That energy enters your bloodstream as millions of circulating glucose molecules, ready to power your body. But there is a biological paradox at play. The glucose freely floating through your veins cannot actually enter your muscle or brain cells on its own. To a passing glucose molecule, the plasma membrane of every cell is a closed system, an impenetrable boundary that keeps the energy stranded in the blood. To cross that barrier, the body relies on a specific signaling molecule, the hormone insulin. Tracing this molecular activation sequence reveals how our bodies extract energy from the world and provides the necessary baseline to understand what happens when the machinery fails. The production of this molecular signal starts in the pancreas. Deep inside the tissue are specialized clusters of endocrine cells called the islets of Langerhans, named for the 19th century pathologist who first identified them. Within these microscopic islands are beta cells. These cells operate as highly sensitive environmental sensors, continuously sampling the bloodstream to monitor your current glucose levels. When blood sugar rises, glucose enters the beta cell and is oxidized to create ATP. This energy spike forces voltage-gated channels to snap open, causing a massive influx of calcium ions into the cell. This calcium surge physically pushes stored insulin vesicles toward the cell membrane, releasing the hormonal message into the bloodstream. The pancreas acts as an active, highly responsive chemical factory, synthesizing and deploying signals in real time to match the exact demands of your environment. Once released, an insulin molecule travels through the circulatory system until it reaches a muscle or fat cell, where it docks into a receptor on the outer surface. This begins the docking process. The insulin receptor spans the plasma membrane, bridging the cell interior and exterior. As this diagram illustrates, when insulin binds at the top, it triggers an internal chain reaction. Following the dashed arrows downward, the signaling cascade activates in sequence. The ultimate target is the oval GSV, a GLUT4 storage vesicle, the doorway for glucose. Preparing a cell for energy requires this intricate biological cascade, where a surface signal must navigate through the cell's densely packed interior. Reaching the GSV presents a severe logistical problem. Because these GLUT4 doors are buried deep inside the cell, they must be physically transported to the outer membrane before any energy can enter. This transport process requires physical construction. This schematic maps out the process. Right in the center is RAC1, a small GTPase protein. Once activated by insulin, it targets cytoskeletal remodeling. Following the arrows down, RAC1 directs the cell to rebuild its actin filament network, constructing temporary tracks to push GLUT4 vesicles outward. Propelled along newly built tracks, the GLUT4 vesicle reaches the cell membrane, fuses seamlessly with the wall, and opens wide to admit circulating blood glucose. Absorbing a single meal requires millions of cells to perform this massive, instantaneous structural reengineering on a microscopic level. Because this machinery requires so many precise sequential steps, it is highly vulnerable to specific points of failure. In type 1 diabetes, the production itself fails. The pancreas stops manufacturing the hormonal signal entirely, leaving the cellular doorways closed because the message was never sent. Type 2 diabetes is a failure of communication. The signal is present, as seen on the right, but the cellular pathway ignores the command. The relays fail. Therefore, the GLUT4 doors remain trapped deep inside. Meanwhile, toxic levels of unused sugar accumulate in the bloodstream. Diabetes manifests as a highly specific mechanical breakdown, either in the production of the body's energy signal or in the transmission of that signal to the cell's interior. High blood sugar is dangerous, but the body also possesses an emergency countermechanism for the opposite threat, running out of fuel. When blood glucose drops too low, a different set of sensors in the pancreas, the alpha cells, deploy a rescue hormone called glucagon. This sine wave chart maps the lifelong biological loop. Glucagon travels to the liver and initiates the release of stored energy, pulling the wave up to keep the brain functioning between meals. Biological homeostasis is an active, continuous pendulum swing. Insulin manages the intake of energy, while glucagon manages the supply. By demystifying the complex biological machinery that processes our food, we can make clearer decisions about our own health and better understand the medical science working to repair it.